Okay, this next video will be talking about the different types of organelles. Once again, membrane-bound organelles has a membrane around it, while non-membrane-bound organelles do not have membranes around it. Let's first begin with the endoplasmic reticulum. This endoplasmic reticulum actually is continuous with the nuclear envelope. So here's the nucleus here, and continuous with the membrane around the nucleus is the rough ER, or rough endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes. So ribosomes are stuck onto it, and what ribosomes do, they make proteins. So the rough endoplasmic reticulum, they make proteins, but what kind of proteins? These are the proteins that are going to be either embedded on the plasma membrane, and you guys should know already what kind of proteins are embedded in the plasma membrane. There's the transmembrane proteins that we talked about, the peripheral protein, the enzymes, or the receptor proteins are going to be embedded on the plasma membrane. They could be utilized for the digestive enzymes within lysosomes. We'll talk about lysosomes in just a second. And it could be exported out of the cell. What is that process when we end up exporting things out of the cell? It's called exocytosis, right? Okay, so these are proteins that are going to be coming out of the cell through the process of exocytosis. Kind of like that neuron with those neurotransmitters that we just talked about. It's made up of cisternes. These are just these interconnected channels here. So these folded areas here are called cisternae. And this is where it ends up budding off to become transport vesicles. So these proteins are going to be coming off of the cisternae and be stored in these transport vesicles. They're called transport vesicles because they be transported to the Golgi apparatus next. Okay. Smooth endoplasmic reticulum. They're smooth because they don't have ribosomes, okay? And because they don't have ribosomes, they cannot make proteins, and ribosomes make proteins. It is continuous with the rough ER, so over here, as we continue on, it becomes the smooth endoplasmic reticulum. And what they do instead, they make lipids, like steroids. They also detoxify alcohol and drugs. So if you think about organs that help detoxify alcohol, like the liver, they will have a lot of smooth endoplasmic reticulum. And also ends up storing certain types of smooth endoplasmic reticulum store calcium. And a specific one that we'll be talking about later in this semester are the muscle cells, skeletal muscle skeletal muscle that will end up storing a lot of calcium for muscle contraction. The Golgi apparatus, as shown over here, it helps modify packages and sorts proteins from the rough ER. So it will also have these interfolded channels here called cisternae. And there's going to be different regions, one that will be facing the rough ER that will take in the vesicle, and then there's a shipping region that will end up after the modification that does on the proteins, it will end up shipping those proteins out. And once again, those proteins that go out of the cell will undergo the process of exocytosis. Those that are going to be embedded on the plasma membrane are going to be just stuck onto the plasma membrane from the vesicles that comes off of the Golgi apparatus. And others will end up becoming lysosomes, like we said, the digestive enzymes within the lysosomes will end up just becoming lysosomes. So the Golgi apparatus actually form lysosomes. And then we said also already the export out the proteins that get exported out of the cell through exocytosis. Lysosomes, these are organelles once again formed by the Golgi apparatus. You see a couple over here. And then they end up getting its enzyme from the rough ER. It helps digest waste products using its enzymes. And that actually helps digest macromolecules into smaller molecules too. Macromolecules, macro means big molecules. So break down bigger molecules into smaller molecules using the digestive enzymes. Once again, what is an enzyme? It is a catalyst. And what does catalyst do? It helps increase the reaction rate of reaction. Okay. And it also helps digest old organelles. So organelles that don't function that well anymore or deteriorating, they help digest that. I had to cross this out, but what autolysis is the cells that will end up undergoing this process called apoptosis. And this ends up programmed cell death, meaning that the cell is going to die off, and so it's going to end up causing itself to die off in a proper way, so it helps prevent damaging neighboring cells. And so autolysis will end up digesting itself using these lysosome enzymes. Another type of membrane-bound organelle is the peroxisome. The peroxisomes are very similar to the lysosomes, but they tend to be a little bit smaller than lysosomes. They are formed by the rough ER though. Okay? And what it does, it helps detoxify harmful substances by using oxygen. So things like hydrogen peroxide is toxic to our cells. But these peroxisome has this enzyme called catalase. Whatever ends with ACE is usually an enzyme. And this catalase, what it does, it will break down hydrogen peroxide into water and oxygen. So it breaks it down to these not so toxic molecules. It also helps detoxify alcohol and drugs as well. What other organelle helps detoxify alcohol and drugs? The smooth endoplasmic reticulum. Okay, the next membrane-bound organelles are the mitochondria, not midichlorians or whatever episode 1 was all about. But mitochondria is a membrane-bound organelle that ends up making ATP. Once again, ATP is our cell's way to utilize energy. It also contains its own circular DNA. So that DNA will ends up allowing it to produce its own proteins, help it replicate itself. If the cell needs to make more ATP, it will end up making more mitochondria. And because of this, there's this endosymbiotic theory where what happened was that these eukaryotic cells end up taking in a prokaryote and ends up having this prokaryote have the symbiotic relationship with it and to make ATP for the eukaryotic cell, while this mitochondria will end up benefiting because it gets energy or glucose molecules to make ATP for the cell. Gets nutrients from the eukaryotic cell, while the prokaryotic cell makes ATP for the eukaryotic cell. It has an outer membrane and an inner membrane. The outer membrane, as you see over here, and the inner membrane, you see how it folds up inside like this. Once again, what does these folds do? They increase the surface area. And in this case scenario, we're not increasing the surface area for absorption. We're increasing the surface area so that we can make more and more ATP. Okay. And this folds are called the cristae. Okay, these folds are called the cristae. There's a space between the membranes called intermembranous space. Inter means between, so the space in between the membranes, and that's along this edge over here. Okay, you'll learn more about this when you go into physiology to talk about how ATP is formed. The matrix is within the inner membrane. So this space, fluid-filled space over here is part of the matrix here. Okay. All right, next is the non-membrane bound organelles. So everything that we covered before were membrane bound. These are non-membrane bound organelles. Ribosomes, you should already know. What does it do? It makes proteins. There's different subunits of ribosomes. There's usually 2 subunits here. And what you see over here is this is DNA over here, this black string thing. And what's happening is transcription is happening. So what happens is that in transcription, DNA is going to be transcribed into, sorry, this is not transcription, this is mRNA over here. I apologize. This is a ribosome over here. The mRNA will be made into amino acid change, will be made into proteins. Okay. So mRNA, this is called translation. mRNA be made into protein. Okay, so they are made up of rRNA. Ribosomes are made up of rRNA and small protein fragments. And there's 2 subunits once again. The ribosomes subunits are made by the nucleolus. The nucleolus is part of the nucleus. It's this part of it. And then it gets shipped out. The subunits get shipped out into the cytoplasm. And those that are going to be fixed on the rough endoplasmic reticulum will end up making proteins that are going to be within the lysosome, be embedded on the plasma membrane, or get exported out of the cell. Those that are not bound to the rough endoplasmic reticulum are going to be just making proteins within the cell. Yeah, we just talked about this, so I'm not going to go over it again. The cytoskeleton are these protein subunits that help make the framework of the cell itself. It helps support the cell, organize the cell, and transport certain materials inside the cell. There are different types of cytoskeletal subunits. One is the microfilaments. Microfilaments are the smallest of the cytoskeletal subunits. They are made of actin. That's a type of protein. And it helps maintain the cell shape. We'll talk more about this actin when we get into the muscle chapter, but it's for muscle contraction. And it helps separate the cells during mitosis or meiosis through the process of cytokinesis. The intermediate filaments are medium-sized, hence the name intermediate. They are made of a different type of protein called keratin. This helps resist stress and helps stabilize certain junctions between cells. Microtubules are the thickest of the cytoskeletal units. It's made up of a protein called tubulin. They'll help hold organelles in place and helps maintain the cell shape to be its normal shape and transports materials within the cell itself. And when we're talking about certain structures like the cilia and flagellum, they are made up of the microtubules, and also these mitotic spindles where they transport the chromosomes from the centrosome. They are also made up of these microtubules. The centrosome, as you can see over here, this only shows you the centrioles, but there's this part that's around it as well. They make up the microtubules that we just talked about within the cytoskeleton. So these centrioles over here are perpendicular from each other, and they will end up having these. They are made up of microtubules themselves. There's 9 sets of these 3 microtubules. And they are replicated during mitosis so that they will end up making these mitotic spindles to help pull the chromosomes away from each other. Okay, so we talked about that. Cilia are these hair-like extensions that you see on the top of these cells here, made up of microtubules like we said earlier. Don't worry about the non-motile one. But those that are motile means that they move. They help project whatever objects that are on top of it. So if there's something on top of this, it'll end up projecting it in one direction. So we have this in our trachea to help move whatever mucus and other dirt particles that are stuck into the mucus to go up the trachea into our pharynx, so we could either spit it out or end up swallowing it. The flagellum, there's only one flagellum, it's only for males. We find it in the sperm, on the sperm, part of the sperm, and it helps the sperm end up moving. The nucleus is once again the control center. It contains DNA, and this has vary amounts of them depending on the different types of cells we're talking about. And like say skeletal muscle cells, there's a lot. Okay, so multiple nuclei. Most cells has just one nucleus. Most cells, but there are some even cells that have no nuclei, and like the red blood cell or erythrocyte. Sorry, erythrocytes. Okay, these are red blood cells. They don't have any nucleus because they used to have nucleus, but they took it away. And we'll talk more about that when we get into the cardiovascular system. Around the nucleus, there's the nuclear envelope. It's a double membrane, and it's once again continuous with the rough ER. And what else is continuous with the rough ER? The smooth ER. So the nuclear envelope also has the nuclear pores. And you guys remember there's these little holes on the pores to allow what to pass through? Those ribosomal subunits, and also mRNA will be able to pass through it as well. And within the nucleus, there's the nucleolus, and what do they make? Those ribosomal subunits that we were just talking about. Okay. Once again, it varies in numbers depending on the cell. All right, and then we're going to continue on to the next chapter and talk about cell cycle.